thought I'd point to a number of the efforts we want to start, even though they're hard to weave together in a simple way. So a big issue is using sorcerer. So we think sorcerer is giving us new insight into the waveforms of cortical sources. So it's not just a matter of making a picture. We want to study the time series of each patch of cortex and how it works. So it's part of our validation study. And we're trying to do this in an organized way that includes analytical tools, but also modeling capabilities. So a lot today is going to be on modeling. But we're starting, for example, with the virtual brain project that you've heard about. We're getting MRIs, but also tractography, the connectivity of the white matter on individual people, and their resting state fMRI, the functional connectivity among regions. So the goal is then our electric analysis, or EEG analysis, can use these pieces of anatomy, these kind of specifications of each person's brain. It's kind of amazing what neuroimaging is giving to us. And if we bring them together, then our mapping of the brain waves can reflect that detail. Mariano is just getting started with using the tractography to constrain the EEG source analysis. We really don't know how to do that, but you know, we're framing the challenge anyway. We want to analyze these source waveforms. So we'll have, at the lowest resolution, 2,400 different channels or waveforms per hemisphere. And Eustace is developing the spectral analysis, the frequency analysis tools, to allow us to look at the oscillations from there, and how certain oscillations relate to others. This is what's called phase amplitude coupling. So the slow waves of sleep, the big giant slow waves when you get into deep sleep, seem to organize the fast ripples of sleep. And that's an example of cross-oscillation coupling, or phase amplitude coupling. And that'll be one of the first things we study with our sleep data. We're getting really excellent 256 channel sleep data now. We want to study those phenomena and build the tools for source waveform analysis. So Eustace is leading the way for that. Roma has agreed to look at some of the modeling tools from the virtual brain. This is the European virtual brain simulation software that can be constructed with the architecture from an individual person's brain. It's kind of cool. We build a neural network simulation, and it's built on the same architecture and backbone as the person's brain. Now, how far we get with that, at the Neuroinformatics Center, we kind of failed over a period of time to make that work very effectively. We're going to try to resurrect it and keep it going. In fact, Sergey has agreed to help us with a grant application to improve the physical simulations of each cortical patch. So how it generates brain waves, we want to build the neural network simulation from this. So there may be a new grant project to model really the architecture that Phan's going over in this cortical column. And you've seen from the feature, each of these neurons will generate a far field, and some more than others. And the whole cortical column taken together with its synchronous activity makes an electrical field that we should be able to measure. How do we understand that? That'll be one more feature of that from it. So the goal really is a kind of an ambitious plan to use modeling and simulations together with real physical measurements in an individual brain. And so you'll be hearing pieces of that, bits and pieces over time. And then, so today, Phan's going to give us a unique bits and pieces, or a bit and piece, from the cortical column and its thalamic control. Is that a fair description, Phan?
Yeah. All
right, take it away.
Okay. So just as, we'll start with a 5-minute review of what we covered last week. So we covered the idea of a column and a circuit within a column. And a column is just a little piece of the cortex that spans all the layers from 1 through 6. And within that, we have excitatory and inhibitory cells. And the cells are arranged not in a undifferentiated mass or tangle of connections between themselves, between excitatory and inhibitory neurons, but very orderly. Orderly in the sense of layers, their distribution across layers, and orderly in the sense of how they're connected to each other. And this is a diagram of that orderliness of connection and layer distribution. And within the idea of a canonical microcircuit is that if you look across all the columns of the cortex, regardless of if it's sensory cortex or limbic cortex, you'll see the same theme, that neurons are connected in this particular way. Last week I showed this without highlighting the red part. We talked about, so the red here, the red connections, the red lines, illustrate the canonical microcircuit. And the canonical microcircuit is not a complete circuit of a cortical column. Rather, it basically emphasizes what we refer to as forward connections. Remember the idea of forward and backward connections. So historically, the canonical microcircuit was defined only for forward excitatory connections. That is, they stimulate a certain input to a cortical column and sort of measured the activity across the layers, and then saw how the excitability spread across the layers. And then use the anatomy of the connectivity to understand how that temporal progression, that is the canonical microcircuit. So let me just remind everybody of what that is. So in the microcircuit, input comes in from the thalamus. It gets presented to layer 4. That's the input layer of the cortex. That's the input layer of the cortex. From there, so you can see here, this line, to a cell, a pyramidal cell within layer 4. Then something happens. That information gets relayed to cells in layers 2 and 3. So this is a forward flow of information within just one column. From this layer, information then is passed down to layers, layer 5 actually. And then layer 5 sends that out to other areas and back up. That's the essential. So, and they verified this, meaning if they record from the different layers, the progression is in that order. What is not traditionally in the microcircuit, or what's not considered in the canonical microcircuit, is this connection, even though it's a forward connection. So the thalamus going to layer 6, okay? And then all these outward projections that aren't to other cortical areas. Back to the thalamus and down to the spinal cord and subcortical areas. And the really interesting thing about this, and it's really important, is that within layer 5, you have two types of pyramidal cells. Those that send their projections out to the spinal cord and the motor areas, and those that are of the type of the canonical microcircuit where it just stays within the cortex and gets relayed to other cortical areas. They're very separate. They don't interact that much. And that's seen here. They're not cross-connected. And they seem to have differential connections with subtypes of interneurons, as illustrated here. That is also true for 6. Layer 6 has projections back out to the thalamus and then projections to other cortical areas. They're also very separate. They don't interact directly with these 2 separate populations in layer 5 as well. So this is really interesting from the perspective of if we think of information just flowing from one cortical area to the next cortical area and gaining complexity as it moves up the cortical hierarchy, what are these other things doing? And why are they so segregated in such an orderly way? If we were to want to model the EEG, want to model brain function, it would behoove us to understand what these circuits are doing. Okay? So that's where we're going to start today, is to try to understand what happens with the outputs in layer 5 that doesn't go to another cortical area, and what happens to the outputs in layer 6 that doesn't go to another cortical area, but down back to subcortex. Okay, subcortex in this case, spinal cord, or the subcortical motor areas mainly, and then the thalamus. Okay. All right. So let me just pause there to see if there are any questions. So that's a recap and then an introduction on what we're going to cover today.
So most of these connections within the column, we think of the column as a highly interconnected unit itself, but what you're telling us is it's segregated within that.
Yeah, yeah, within one single column, which if you're talking about the size, this is micrometers. Yeah?
Yep.
You have what seems to be 2 to 4 independent pathways.
Yep.
Yeah.
And why do they connect back with the thalamus? I thought the data already came in. Why does the cortex have to communicate back again?
Yeah, that's the core question. That is a core question. All right. So, and it bears to say, or to be emphasized from last week, cortical connections or neuronal connections, both at the thalamus and at the cortex, can be or the influence of a cell on another cell in terms of this connection, particularly excitatory, can be classified to 2 groups. One is driving, that is if a cell is active and is connected to another cell, its activity is strong enough to activate the other cell, to drive the other cell. That is typical of a forward connection. So from the thalamus to layer 4, it's a driving input. Other types of responses, another type is what is called modulatory. It's not strong enough to drive. It seems to only modulate, and some of the mechanism is related to opening the ions channel directly, which is ionotropic. And then the other is a much slower process, which is metabotropic, which requires some other processes to occur within the cell, and then it opens up those channels. So the modulatory are of the latter type, that it's just delayed and it's not. And so that's a really important distinction to make within all what we're going to discuss here. So here is a diagram by Sherman and Guillory. These guys study the thalamus. And they ask this exact question. What is the role of the circuit in layer 5 that goes out to the pyramidal tract, or the motor areas of the brain. And what they conclude is actually, to me, quite profound. So let's start with this. The main idea is this. You have sensory information going to the cortex. Something happens, it does some transformation happens. Part of that information goes to another cortical area. Part of that information goes back down to the motor areas. And that's this output here in layer 5, back out, okay? And you can see here, motor outputs, all right? Here, at this level, these are the nuclei of the thalamus. So let's take the case of visual inputs. So visual input comes from the retina, goes to the lateral geniculate nucleus, the primary nucleus, that just relays information about what happened with the retinal ganglion cells. That's all it does. It sends it to 4. The cortex does other things to it. From the same axon, the same ganglion axon of the retinal cells, it branches. It goes to a motor center within the brain stem. That's the superior colliculus and other areas that control head and eye movements. Okay? The same signal, a sensory signal, forks and goes to a motor center. So it's not conveying something different, it's conveying the same information. Now, this is the central idea that Sherman and Guillory proposed, that in the course of evolutionary development, a sense organ would have no place if it couldn't influence some kind of motor output. It makes no sense. Okay? So, the first thing then is it goes out to the motor area. We'll talk about what then is the implication of the motor in just a moment. But what I want to do is then trace this now to, let's just say this is V1, the primary visual cortex. From there, information, when it goes back out, the same neuron in layer 5, these are the big pyramidal neurons that have long axons that go out to the cortical areas. It branches. Just like the incoming one, it branches. It sends information to the secondary thalamic nucleus. At the same time, it sends a branch to the motor tract. And then it repeats that again and again.
And this is tactile again. So this is not motor cortex, this is midbrain.
Yeah.
So it's the retinotactile pathway and then the midbrain pathway for orienting attention.
Head movements, eye movements, pupil dilation. Basically, if you're talking about the visual modality, basically orienting to where you're going next, or to the visual field. But then it repeats this over and over. Okay, so one stream, that's this. So we can think about it, incoming information in the feed-forward way. This goes to another cortical area. And then this here, the pyramidal tract, goes back out, back down to the motor center. Okay?
And these higher order, there are multiple higher order thalamic nuclei.
Yeah.
Each HO is another one.
Yeah. And so that's right. So here's the interesting thing. Layer 5, remember the canonical part of the microcircuit. That basically what it means is you can look at any area and this structure will hold. So the big pyramidals that go out to the motor areas are there in V1, where it has traditionally, you don't think it should be connected to the motor area. You go to V2, it's connected to motor area. You go down all the way to limbic, it's connected to motor in some form, right? So that's what's powerful about this idea of a canonical circuit, okay? Because you can just say, all right, to understand other areas, I only have to know this kind of layout. Okay. And then, so what they said is this. The function of parameter 5 output is to relay information, very much like the primary is to relay information, okay? But what is it relaying is the question. What is it relaying? And what are sort of the consequences of passing through this? What happens when it's relaying? What does it do to the information as it relays? And here they pointed out that they didn't discuss what 6 does. So you can see here, 6 then also projects back down. It doesn't project forward. It projects back down to its original site. But the dashed line is modulatory. It's not driving, okay? So we'll talk about that in the second half of the session today. So the profound thing here is they say that it's essentially relaying information about what to expect. So the output of 5 is what to expect to occur in the sensory environment as a consequence of a motor response. And the hard to grasp aspect of this is, why is it so early? Why is it in V1? But this is where they go back to their reasoning. That no matter where you look in the vertebrate anatomy, you see this branch. It's one single piece of information that goes to two places. So they're saying that in the case of V1, when the incoming visual input, it sends it to the superior colliculus because it's saying, well, you saw this, you're going to be prepared to see something a little different based on the consequence of some action, like a little saccade. Because one of the problems in neuroscience is, how do we perceive things as stationary, for example, when we're moving our head? How do we make that distinction? The idea is that whenever you make a motor command, you send a signal to the areas that will receive the sensory input about what the plan, what the action is. This is called an efferent copy, to prepare the sensory input to know that it is the change or whatever you perceive is caused by you, not by something outside. But they're saying this is as early as the first sense that you get.
Because this efferent copy was a motor copy. So your motor plan informed your sensory brain, look out, I'm moving right.
The tradition.
But now you're saying that the first perception is already tuning up the motor system for how to pay attention to it or how to act on it?
So in the 40s and 50s, people were already thinking about, or even earlier than that, this idea of an efferent copy. But it was always thought that it happened, as Don said, when you have an articulate, a motor plan in the motor cortex, or the frontal lobes at the very least. When these guys, Sherman and Guillory, looked at this very fact that a branch of one signal goes to two places, they said this is basically unavoidable, this idea that perception is embodied, that it is not independent at any level from the motor, because the most primitive is the reflex arc, right? You don't even need the brain. You recoil from a hot object or something like that. Sense and motor are tied. This is just an elaboration of that. So the question is, how do we carry that from layer 5 from one area to the next? So what they're saying is that you have from this area a motor copy, and the distinction here becomes a little philosophical, because they admitted that part of this is we haven't built up the body of evidence to prove any of this yet, because people haven't tried to look, because they've never even thought of it in this framework. But there are data that are consistent with this. But the anatomy is not in dispute. The anatomy says we should be looking for these things. So their idea is that essentially what you're doing is this is relaying that efferent copy to layer 4. Okay? And remember, layer 4 here, right, higher upstream, is receiving information that is already processed by the other layer, so it converges into the same place. And so there are consequences of this, of course, because they say what it does is it sort of gates. So it's gating the flow of information based on your state, whether you're moving or not, is just an example of one gate of how an efferent copy could affect that. So one of the things is when you look at an animal and it's in a restful state and it's not moving, the cells in the secondary thalamus sort of just is in this mode of tonic firing. But when all of a sudden it starts to move, or you're asking it to do a task, it switches into this mode of bursting. That when input comes in, it bursts. And this burst is sort of like a very loud signal. So it's a loud signal that says that is received by these cells in the upper stream that is can activate them much more efficiently. So they interpret it as the efferent copy in this way. They interpret it as the efferent copy is nothing. Don't expect anything. So its influence on the upstream is not that big. But all of a sudden, if something unexpected happened that is not part of your efferent copy, then all of a sudden it sort of primes the other area to be. They call it. They describe it as being analogous to a wake-up call. We'll talk about how that how that can play out.
Let me just ask about the architecture here. So you're saying we have multiple cortical levels. We have primary, unimodal association, heteromodal, and then limbic. You're saying each one has its own thalamus nucleus?
No, not each one. Good question.
Could be the same HO there, or is it different HOs?
So here, different components. So here is, maybe you saw this in your reading, you're just setting me up.
No, sir. This is a naive question.
So this is data that, so this is the superior colliculus. Again, it's just a brainstem nucleus that controls eye movements, among other functions, but its motor function is eye movements. So you can see here, so it's layered, and it's columnar. You can see when you trace the pyramidal outputs to this, it is divided in this way. Area 17, 18, so 17 is primary, area 18 is secondary, V2, V3, and so on. you can see that it goes back to the superior colliculus, but it does so not in the same position. And if you go through to area 4, the limbic cortex, and so on, you can see that it goes deeper and deeper, this distribution, within one motor structure. So their argument is this: the farther you are away from, or the earlier in the sensory processing stream, you have some kind of a motor plan, but it's not, again, they say, don't think of the motor plan as something that's articulated coming from the motor cortex. It's just an abstraction, really an abstract idea based on that forking of the same neuron, the branching, that it's got to be some kind of information about motor. And they say that as you go deeper and deeper in this, the farther away you go from sensory and this structure, and I don't know much about the superior colliculus, I guess the closer you are to the bottom layers, the more likely you are to influence an output of motor. So I take this as not that the HOs, the higher order, these are distinct, nuclei. But there are so many different motor centers or motor nuclei or structures that can influence motor subcortically, that this is more of a simple way to organize your thinking about it. But generally, in the somatosensory cortex, for example, it won't be the LGN, the lateral geniculate nucleus. It would be the ventromedial nucleus. And then the secondary one is the posterior nucleus. My guess is within that, you'll have some kind of a finer differentiation so that the HO themselves are sort of organized somewhat similar to the superior colliculus, but the fields should be separable. Okay. So far, I mean, to me, this is sort of everything that we talked about is sort of counterintuitive, right, to everything we've learned about the separability of information and construction of meaning, or making, extracting some information from the sensory inputs. It's so counterintuitive, but very cool. All right. So this is a very recent paper, 2019, where they're looking at exactly this issue. What's going on here? Okay. So as information from 5 goes to here, goes back up, what is happening? And this is also a good chance to review some topics we discussed last week as well. So here what they did was they injected, they wanted to ask, so this is the posterior nucleus of, the ventral posterior nucleus of the thalamus. That's the secondary somatosensory thalamus nuclei of the thalamus, nucleus of the thalamus. So its position would be somewhere here. And so we know that primary motor cortex, so this is S1, okay? This is M1, and this is S2. So motor cortex, primary motor cortex is directly connected to primary motor cortex. Secondary, somatosensory, sorry, what did I say? Yeah, so this is S1, primary somatosensory, primary motor, secondary somatosensory. And so they wanted to see how these are connected to primary sensory so that they can start understanding the influence.
So now these are, I don't understand these connections, because normally there is a topography of connections that you have in the upper right, from primary sensory or motor to unimodal to heteromodal to limbic. We have an organized architecture, but now we know that primary motor is connected to primary somatosensory, but what are those connections like? Are they point to point, 4 to 4 or 2, 3 to 2?
I'm going to show you right now.
All
Yeah,
right.
this is why I wanted to bring these two figures up from last week, right? So let's take the connection from the palm, the thalamus, to S1. Remember last time what we said was this is the core. This is if it was the LGN coming in. This is the matrix. Usually, some aspects of LGN is matrix, but as you move away from the primary relay nuclei, they become more matrix-like. And remember, there are different protein bindings that define these cells, these line of cells. So here is the model, right? This is the matrix connects to layer 1 and layer 5A. That's exactly what you see here. So layer 1 is here, so it's very intense in its color. This is agranular, right? M1 is agranular to eulaminate, right? So it has this structure, right? It goes to very superficial. Again, that's what you see right here. So if they were to inject a tracer here, see where it ends up in primary somatosensory cortex is here, layer 1. You see that? Very consistent with this. This also predicts layer 5. This is layer 5. Okay. There's some layer 6, of course. And then S2 would follow this pattern, right? This granular or less granular to more granular. So S1 to S2 would follow this. It would be distributed in layer 1, which you do see, and some in 4 and 5. But here it's mainly 5, and then a little bit of 6. So and so the palm here is here. Not in 4, you see here. So these are the whisker barrels themselves. So if you were to look at the representation of one whisker, the input would be around here, and it's absent there. Some in 5, okay? So it more or less follows this sort of pattern, both of the structural model of connectivity for the cortex and the thalamus, all right? So they did something interesting. So now they know how things are connected. So they use optogenetics. And what they did was they can activate each one of these axons, whether it's from the thalamus, from M1, or from S2. And then they record a cell or parameters in layer 2 and 3. After they stimulate. And this is an example of this. Here is for when they just activate the 2-3 neurons by just stimulating the whiskers. So you see this response here. Okay, so this is optogenetic pulse, light pulse, very short. This is when they are activating with the posterior nucleus, okay? And then both at the same time. So you can see that when you activate both at the same time, it is greater than each one individually. So it's consistent with this idea that the information from the palm goes to layer 4 and then enhances its firing. So it's producing a gain in that layer.
Priming.
Yeah. But you can see that that's not the case for M1 or S2 because they're also sending, you can stimulate those, but they don't have an influence on layer cells 2, 3. So this is actually pretty remarkable. The other thing that they show is that here is this initial peak of activation, and there's the light pulse. It depresses, but only when you have input from the palm that you then get a rebound in sustained activity. And this is it right here. And so you can see that right here. These little gray traces are just individual parameters firing multiple times and average for that, and then this is the population average. So you can see this is, they say this is in alpha beta frequency. So essentially, and this is, so then they trace it back, is it, it's something that's happening, this here, is it happening at the dendrites? Is it part of the 2-3, or is it the actual influence from the thalamic nucleus? And they say it's actually in the thalamic nucleus. So it's that. About a year ago, we also talked about the thalamus' influence in sustaining oscillations. This is very consistent with that. The idea here is this is now some kind of an activation state, and they even say this is very similar to a spindle. The mechanisms are very similar to spindles, and so this is for somatosensory cortex. They say it's also observed when you look in the visual domain. In the visual domain, the pulvinar is the secondary, and it shows the same kind of behavior. So it's a relay, a secondary relay that has this sort of oscillatory gain amplifying. But remember, don't lose sight of the nature of this input. The nature of this input is an efferent copy of what to expect. So it's almost like a state of expectancy. Yeah? So it happens very early.
So it stabilizes the circuitry for a period of time by that oscillation.
Yeah,
is that right?
yeah. Or extends sort of the time window.
Yeah.
Of information integration by 2-3.
This is a function of the matrix projections of the thalamus.
Well, they talked about what possible ways can it do this that is different than, say, remember these guys sort of give similar inputs, right? Because it's superficial. So one mechanism of action they speculated is the different. So layer 1 doesn't have any excitatory cell body. It's cell sparse. What it does have are a lot of axons of other dendrites of incoming cells from other areas, dendrites of cells from the same column, and inhibitory interneurons in that layer. One proposal, a mechanism that they proposed that could distinguish between the three is the type of interneurons that it's affecting. And they say maybe it's the serotonergic ones that allows them to differentially sustain this activity this year. Okay. So now the power of this model, of course, is that you can try to think about things abstractly as it's moving up the hierarchy. So what they say is that as you move away from the sensory areas, information is getting more complex. Just like early sensory areas, you only are sensitive to line orientations. And then you have bigger receptive fields so that it covers more orientations and so on. So that is sort of a building up of sensory information. Simultaneous to this is the building up of motor plans across this hierarchy. Simultaneous, not wait until you understand fully what it is. And think about this for memories, right? Because the traces of memory are distributed once they become consolidated. You can't you can't separate the two. And indeed, that's what we see when you go up to higher sensory areas, like parietal cortex. The motor action is inseparable, right? If you understand the concept of a fork, you know what the motor action that you can engage with the fork. This is sort of affordances at the very core, meaning that an object gives you or tells you what to do, or inherent to that object is
What
an
to do
action,
with it.
what to do with it. And so this is just a breaking down of that whole concept to a very sort of the atoms of it, even, right? You
No.
know. All right.
And I would say, we never thought, when we could do this in the cortex, but we never thought the thalamus would play this role of kind of integrating the multiple levels from the very beginning. It's almost like a cascade, that the implications for action start at the first level.
Yeah.
And the thalamus is setting up those relays, even though it's a really dumb architecture, it doesn't have any concepts, it's just relays.
Right.
But they prime the next level in this kind of ordered way.
Right. The other is, oh, go ahead.
So for me, it's still this idea very non-intuitive. If speaking about fork, why would my motor output care about the directionality of fork? Whether when it gets input from V1 or V2, rather than it should get the input from an object in the fork itself, per se. That's where my intuition.
Yeah, I see if I understand it. You know that why would it matter until you actually get the concept of a fork?
Object.
Yeah. Well, that's a good question. I think the fallback here is if we don't have the concept of a motor plan, an efference copy, how do we understand this fork? This splitting of the same information, because it's not. So here, it's just one signal of the senses and the reflex arc, and this is the part that I found hard to understand is what is that action? What is that efferent copy? Because phenomenologically, right? I mean, your question is sort of like a high-level phenomenological experience. You know that you in your everyday experience, a fork only has meaning as a fork. You know that as an object, the whole object.
Once it's realized. Yeah. Well, I think the other thing is to look at the nature of these midbrain functions. So the midbrain is a very primitive area. Now we need it. Remember the studies, Weisskranz studies of blind sight. So patients with cortical lesions couldn't see something, but they knew it was there. So you give them an object, and they say, I don't see that, but just guess where it is, and they would tell. And it was their midbrain that was doing that kind of a function. So I think what's happening here is not the fully understood fork. This is eye movements and head movements that are saying, uh-oh, there's something over here.
Yeah.
It's like the frogs, bug detectors. They're very primitive, reflex-like, but it's going on from the very beginning of perception. The first coming into V1 is saying, uh-oh, there's something to orient to over here.
Yeah.
Maybe you should orient. But it's only the first clue. So you probably don't have an orientation or an eye movement or head movement until you get up to the third level or, it accumulates.
Yeah, and I have 10 minutes to cover the next one, which is layer 6, but and so, your point too is because when we see things in the environment, we don't just see a line, we see the whole. That's the level of consciousness of awareness that we have. So you can think of motor actions as that way too. That we don't experience the atoms of it. We experience the whole grass, but the parallel line has to have some equivalent the orientation line has to have some equivalent of a motor. Is all you can say from this until yeah. Okay. Oh, and the other thing. Yes.
I think perhaps one way to look at this is this provides, as you go up in level of analysis, it kind of provides an increasingly articulated bias in terms of your motor output and your expectations of what to do next. But it's very important that bias may be wrong. And so I think the efferent copy all the way along always provides you with a fallback in case something goes wrong, perhaps. And so it kind of layers this increasing articulation with a copy of what's happening at each level of bias going along.
That's right. It provides that, but so the next level up takes that efferent copy and does a comparison. So they actually say this is a tripartite model, not a bipartite. That it's sensory, it's an afferent copy, and then a reafferent, meaning a comparison. So it conveys So the
there
next
is discrepancy or comparison.
Yeah. And then the other, of course, this is a simplistic, right? There are many other motor centers, other inputs to this that may be motor related and brainstem related, arousal related, and so on, that could influence, again, the gating of that efferent copy, and that's really important. This is only a simplified. All right, this next one. So, all right, if that's what this circuit here is, again, separable. Yellow is the pyramidal from the intracortical. And then down in layer 6, you also have the separation of the thalamus to the intracortical. So how do we understand that? I'm going to take 5 minutes, and we can talk about this more in future sessions. But we've covered this before, actually. This is the Grossberg's, Stephen Grossberg's model of what would he call adaptive resonance theory. And the particular mechanism of adaptive resonance theory is this idea of a folding feedback. Okay? This is like, he tries to explain why don't we have hallucinations and so on. We don't have time to go into that, but this is really important. And remember, the other important thing is this is modulatory, okay? This is modulatory. This is not driving. So we can look at and I appreciate this more now. When I presented this before, I didn't understand the split in layers 6 and 5 of these two parallel pathways. This one is specific to this output. It's not here. This is not the cortical. This is this structure using just the thalamic outputs. So input comes in, okay, like we noted. It goes to 4, and then it goes to 6. In 6, it goes up to 4. We know that anatomy as well, or that circuit. But here he adds the interneurons, okay? And this is what makes it modulatory, according to him, not the inherent fibers or anything like that. But there is a reason why there's a fiber specific fiber differences. That's the anatomical basis of the differentiation of the two classes. But you can see here, this is an excitatory neuron. These are inhibitory neurons. So the LGN drives this. This essentially provides a center on, surround, off. So you can see that it touches on these inhibitory neurons, shuts these guys down. And this basically, because they're more inputs excitatory than inhibitory, this is the on. Okay, so this is the basic idea of center on, surround, off.
Of a feed-forward circuit.
Of a feed-forward
This is the
circuit.
input data coming in setting up a pattern.
That's right.
Like a receptive field.
That's right, okay? And then, so what you have then is now that V2 feedback to V1. Remember, it goes into layer 1, right? We talked about that. S2 to S1 would be to layer 1 as well. And layer 1 is doesn't have a lot of cell bodies, so you can see it here. Cell in layer 5 reaches up to here. Okay? So prior information goes back down, 5 passes it on to 6, and then it loops back up. Okay? The importance of this is why he calls it folded feedback is these are this is a mechanism of attention or higher order influence. So this is basically, to describe it psychologically, this is what to expect. Okay? It goes back down to 6. Okay? So incoming information then, has to match up. So here, if the LGN is coming up, and then you have this coming back down, it's saying what is coming in has to match up with what you expect. Okay? So that's the folded feedback. So that's attention. If you expect something to happen, you're faster to recognize it when it happens, as an example.
So this is V2 layer 6
Yeah.
into, where's it going? Layer 6 of V1?
No, layer 1 of V1.
Oh, I see.
And
Then
then-
layer 1's part of that
Yeah.
circuit. Got it.
So it's consistent with this anatomy here.
Yeah.
You know, so this is this cell right here, this cortical cell.
Yeah,
If I drew the apical dendrite, would go all the way up.
right.
Yeah.
That's where it's getting.
Yeah. And then it's also consistent with this anatomy here, the thalamic right here, and this going up to 4. So this is just isolated for that one circuit. And then and then the second component of adaptive resonance theory is layer 2, 3. It's not anything different. He uses the term grouping. Basically, you can think of it as if incoming information is processed and represented, layers 2 and 3 form some kind of a population coherence. That's just a group. And so what he says is that that this goes up, okay, and either turns on or turns off this, and allows the lateral connections to have an influence to form these groups. So it's sort of like, once it's confirmed, it feeds back into the representation in the upper layers. That's essentially it. The final thing here is 6 goes back and influences the relay. And that is
In the thalamus.
In the thalamus, in its primary. So not only is it influencing the feed forward, but it's also influencing what's coming in. This is, to him, attention. This is why you need a model of the world to make sense of things coming in. But this is the same mechanism of why, when you expect something, you don't hallucinate. Right? It has to match up. It has to resonate. It has, and that's the adaptive resonance, is that it's always sort of matching your internal expectations with actual external inputs. And so this is just a high level of it. So now I want to revisit this really quick. To now sort of understand the full layer implication of a column and the canonical microcircuit. Maybe we'll leave that for the next couple of seminars. And then even think about then, within this, what are the rhythms that we're talking about, right? I mean, I tried that before, but I think I have a better place to start to understand the mechanism, particularly the thalamic influence on the cortical oscillations. But also, of course, if we were to build a neural model of this, what would we expect each one of these circuits within a column to convey in terms of information. So I'll leave it at that.
Very good, right on time. That's great. So the readings we should be looking at, Gilroy and Sherman, and you'd also recommend the Grossberg?
Yeah, it's always good to go back, even if you've read it,
No.
just because it's a slightly new perspective. I mean, I didn't appreciate most of that stuff until I convinced myself that there are these two parallel circuits in 5 and 6. Because the 4, 3, 1, and back down sort of makes sense. You don't have, doesn't challenge your sort of intuitive understanding of the flow of information. Yeah.
What did you, what exactly did you say they believed was serotonergic?
How the secondary nucleus or nuclei of the thalamus can have a differential effect on 2-3 processing when M1 and S2, as an example, project to layer 1, which should influence 2-3 as well in the lower areas.
Mm-hmm. I was wondering if that might be related to attentional and kind of motor effects of depression.
Interesting. Well, certainly, yeah, a lot of this, it's like you can't talk about this now without invoking higher order, top down, i.e. attention. I mean, this is much more abstract than a little more abstract than the cognitive scientists and, concept of attention, but it is attention in its flavor. But depression I haven't thought about.
I'm just thinking in terms of motor slowing, but more motor slowing by bogging down this whole feedback mechanism and expectation.
Yeah, no, and think about this. Entertain me for 2 minutes as I try to answer Kathy's question. This right here, you can say, see, persistent activity. This is, if we were to think of the secondary nucleus influence as sort of like an attention, meaning that it now all of a sudden it extended the window of processing. This is essentially what it's doing in the neurophysiology, is that it's making these cells active for that period of time. So that could be.
Would this be like the beta influence?
This is
to
beta.
Stabilize the motor
This-
engram?
Exactly. So this is beta. So essentially, what it's saying is that influence is a stabilizing influence for information processing. Now, take the case of what we think beta means in Parkinson's. One of the theory of what beta is, it's a stabilization of the motor. Well, maybe that's just another way to say that it's attention, right? Keeping things stable, and the inability then is to switch your attention, to break up this. So attention could be good, but maybe you can see that it's bad if you're unable to disengage your attention. So a motor plan, a steady motor plan, is sort of like an attentional aspect of the motor. If you can't switch it, then you have things like Parkinson, the slowness of movement, the inability to switch. So this is exactly that, yeah. Not that how people would explain it, but that's how I would understand it.
So this might give us clues where to start looking, or at least how to think about the neurophysiology. I mean, we have to look in the cortex.
Yeah.
That's the main thing we can see with the EEG. But you're saying that some of the oscillatory features, let's say a phase amplitude coupling in beta, might be attributable to these secondary thalamic influences.
Yeah.
Well, what about the striatum? I thought that's where the dopamine modulated beta influence comes from.
We should take the basal ganglia as a separate topic, because I think it can be understood within this framework too, because we take the structural model of connectivity. We should be able to reason, because 5 goes out to the basal ganglia. Layer 5 goes out. That's the output layer to the inputs of the basal ganglia. When the basal ganglia comes back out, or it's feed this output through the thalamus back out, it follows the same structural connectivity, layer 1 and layer 5. We should be able to reason about it. So I think that's a separate topic, but using this exact same structural model.
Well, I think this would be really great if we could go through this systematic, look at the Parkinsonian effects, look at the classical models, but then start breaking it up.
Yeah, but Kathy's question is actually quite interesting for sort of the motor retardation and depression as well.
Yeah.
As a slightly different note, Phan, in response to your task switching comment, there's a lot of literature that directs impairment with executive function, specifically set switching, and attentional commands like that are related to postural instabilities, balance, and aging, but particularly in Parkinson's.
Yeah.
So they've identified pretty strong links between.
Yeah, I mean, you can think about postural instability is the inability to switch between the fine motor control to maintain posture.
Exactly.
So that rigidity, that fixedness, is the adaptive brain is a switchable brain. You have to be able to switch.
Yeah.
Cool. So there'd be analogous cognitive defects?
Yeah, there's very strong just correlation studies looking at association between cognitive function, impairment, and then postural control.
Yeah.
But there's strong links between the two. And both the more just simple aging, so maybe mild cognitive impairment, pre-Alzheimer's, but also specifically in the balance impaired movement disorders, Parkinson's group as well. We see a lot of tasks.
I mean, there's always the danger of this being too broadly, of a framework, and then you can fit everything.
Yeah.
But it's not unreasonable, I mean, to the extent that we can think about reason through the basal ganglia as well, because I think that is so critical, because there's so many of these basal ganglia loops, but they must follow the same principle of the structural connectivity.
Isn't context switching a difficulty for Alzheimer's patients too?
Yes.
Or lack of, inability to focus?
Yes, but I think there's possibly different root cause. But I know that set switching particularly, or executive functions in general, are very closely looped with postural control or postural instabilities. There's a pretty big literature there. It's very interesting.
Yeah, that's important.
So if you could improve postural control, could you improve their set switching?
I don't know if it does that.
That would be a good hypothesis.
That would be.
I mean, if
If the mechanisms are similar.
yeah, that's a good hypothesis.
And physical training, physical therapy is definitely protective of cognitive functions. But I know people are looking into that and trying to understand the causal effect and the directionality of that. But
Yeah.
I'm not
And
sure.
I'll bet, if you look at the literature on Parkinson's with even dopaminergic therapy, there's probably a correlation between motor improvement and cognitive improvement. There's got.
And what about the dyskinesias, though?
What
You
is
might
that?
think the dopamine is causing damage to cognition, just like it does to
I
motor.
would say so too. That's
I mean, it makes sense. I mean, look at what exercise does to people's moods.
Right,
It's
Yep.
just,
right.
there's
Yep.
always that connection.
Yep. Or you can even say, as I was reading this, and I got cognitively excited. I couldn't sit still either. I needed that. So it goes both ways.